Hello students. So in the previous video I told you about the random sampling, and we have calculated some the species frequency, and then I was explaining about the species density. But just a brief review. Random sampling means when you are going to collect some samples from a huge population which is evenly distributed. I mean, there is no selection pressure, and all the organisms in that particular area are equally interacting with the environment. So it's up to you from where you have to collect the sample. It does not matter that you have to select organism for a particular place. But in systematic sampling, there might be some factors which affect the distribution of the organisms in the population or in the community or in a habitat. So care should be taken while doing sampling, because you cannot take random sampling. There are some factors which favor some organisms as compared to the other one. So if you are collecting samples from the center of a field and there is some river at the banks, at the edges of the field, there might be some plants having large proportion at the edges of the field, not in the center, because one of the factor which is flowing of water affecting it. So in this case you have to take the systematic sampling, which we'll discuss later on. But as we were discussing about the random sampling in the previous class, and I told you that if you have a quadrate, we discussed about the quadrate method, because when you're talking about the random sampling, we have two types of methods to calculate the biodiversity. The random sampling you have to do in two ways. One is called quadrate method, and the other one is called percentage cover. In the quadrate methods we have discussed, you can get more information about the quadrate in the previous video. Here we are placing some quadrate in two different communities. Like we just have to calculate about the two types of species in that different communities. Here is the species. We just select only two species at this time for simplicity. So species A and species B only. So let's see. And we just have quadrates, like we have so many quadrates here. This one, here. So here are the quadrates, number of quadrates we are putting here. So 1, 2, 3, 4, 5, 6, and let's make them more. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8, 9. And yes, we just make one more, 10. So this is quadrate 1, quadrate 2, quadrate 3, quadrate 4, quadrate 5, 6, 7, 8, 9, 10. And we just looking the species A and B in two different in the quadrates, in which quadrate which species is existing. So here this is species A were 5 and species B were 7, 3, 9, 6, 2, 5, 3, 6, 7, not 2, 1, 3, 5, 6, 0, 2, 1, and 5. Now these are the number of individuals of each species in each quadrate. Like in quadrate 1, 5 individuals of species A are present and 7 individuals of species B were present in quadrate 1. In quadrant 2, 3 individuals of species A and 9 individuals of species B. Similarly, 6, 2, 5, 3, 6, 7, 0. So, now we just want to calculate the species frequency. How can we calculate the species frequency? So, species frequency. How can we calculate it now? The formula for species frequency is now the existence of species. I mean, the more better option is to number of quadrates possess species A upon total number of quadrates. And then it should be taken into percentage multiplied by 100 as well. So if you want to calculate the species frequency of species A, so there is 1, 2, 3, 4, 5, 6. There are 2 quadrates where species A does not exist. Species A is not present in quadrate 6 and in quadrate 9, but it is present in 8 quadrates. So we say that it should be 8 upon 10 multiplied by 100. That is equal to 80%, which is the species frequency of species A. With the same formula, if you see here, all the quadrates have species B, so species B is 100%. This is for species A and this is for species B. Now we have calculated the frequency of species in the quadrates. Right, now it is very simple, but what does it tell us? What does it tell us? It tells us that how much these 2 species are distributed in an area. The higher the percentage, the higher the frequency, the more evenly the species should be distributed throughout the area. It is not concerned with the number, abundance, density, how many individuals are present. Suppose if I'm saying that, like, here is a field of a particular area, and a species is more proportion, but it is only present at one corner of this field, a largest number of individual of a species. But its distribution is quite less because it doesn't exist here, 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 here. It's not present anywhere, but it's only present at one corner of this field. So its species frequency is quite low. But if there is another species and it is only in quite less number, but it is distributed like in this way, so we say that its species frequency of this black dot one, if this black for X and this red for Y, then we say that species frequency of X is quite greater than species Y because it's randomly distributed throughout the area, not concerned to its number. This is all about the species frequency. Now what about the species density? When the second, when we calculate in the quadrate method, so we have, 2 important things in the quadrate methods. One we have done like the species frequency, and the other one which we are going to calculate now is the species density. We have calculated species frequency, and I told you about it. Now what is species density? Species density basically depends upon the total abundance of organisms in each species. And I told you in the previous video about the biodiversity, what is abundance. So abundance, if you count individual of each species, A, and their summation will make up a total abundance. Like here, 5 plus 3, 8, then 14, 19, 25, 26, 31, and 32. So the summation is here 32, which is the total abundance of species A. And here 16, 18, 21, 28, 30, 33, 39, 41, and 46. So summation is 46 here, total abundance. Now we want to calculate the species density, how much a species is present in a particular, in a unit of area. In the same way, like you just have to compare mass with density. So what is the density? Density is also mass, but it's a per unit volume. Here in the same case, we say species density. Species density means also total number of individuals, but that not total number of individuals. The number of individuals in per unit area is called species density. So if you want to calculate species density, what should we do? 32 individual of species A for 10 quadrates. If one quadrate have one quadrate have an area of 1 meter square. So 10 quadrate means 10 meters square. So in 10 meter square area, we have 32 organisms. So if you want to calculate the species density, what should you do? You just have to divide 32 by 10, which is the meter square, 10 meters square, because one quadrate is 1 meter square, so that's mean 10 meters square. So it means it's 3.2 individuals per meter square. That is the species density. So its density refers to the abundance of organism in a particular area. Like if you see, once again look at this field, this red one, the Y one, has more species density because it's in per unit area is quite richly located, and they are so far away from one another, but they have to cover up a lot of area. So they have their own importances, because when you are talking about the distribution, it means the random and even distributions of a species means that there is a little changes in the surrounding, in the environment. The environment is quite stable and a species is not stressed, so it can survive everywhere in a community or in a habitat. But if there's very less frequency, it means there's some stressful condition in some part of the community or in some area where these organisms cannot be well adopted. So they just have to survive in a particular area, not in the entire area, because of the some pressure, maybe the predators, maybe some diseases, maybe some catastrophic situation, something maybe. So this is one of the case, the differences between the species frequency and species density. So these two methods and these two terms can use in the quadrate methods. We can apply quadrate to calculate these two features of the random sampling. Now, sometimes it is very difficult to count individual organism in each quadrate. You are not able to count because sometimes the organisms are so quite tiny, but they are so branches. They are so dispersed, so branch out. I mean, like here is, I'm just giving you an example, like here is a quadrate. Here is a quadrate. In this quadrate we have squares, and we just calculate the organisms, how many in number. Like here is one of the organisms quite tiny, but it's branch out, trailing like grasses, and it's very difficult to count this organism. How many organisms should, maybe there is an error that we had to count. We may count this one, all of these are the branches. They are not the individual organism of the population, but we just also count this as a individual plant. So in some cases it's very difficult. It's not able to differentiate between either they are belonging to the same organism, have branches, or they are a unique organism. So in this case it's very difficult to count them. We just then count the number of these squares, because one squares, if this is whole 1 meter square and we have 100 boxes, then it means each box has to be 10 centimeter square. So if you just count them, the number of boxes, so we just give like one box we give as 1%. So how many boxes this plant cover? Let's suppose 1, 2, 3, 4, 5, 6. So it's not completely, but half of them. So we can also calculate as 0.5. Like just for example, we can say of a plant cover 2 quadrates, then 4 quadrates, then 3.5 quadrates, then 6 quadrates. So the total should be 6, 9.5 and 15.5%. It should be calculated into percent, and this is called percentage cover. So they cover such area because we cannot count the individuals in each quadrate. So sometimes we use the percentage cover. Now, percentage cover also have a scale which is called the abundance scale. What is meant by abundance scale? Abundance scale means it's like the international scale calibrated by the ecologist, biologist, and they say that we have to give certain types of numbers to represent the percentages. Numbers like in binomial number, you have just only 2 numbers. You cannot have any third number, fourth number. In this nomenclature, we have just only 5 numbers. And we say if a scientist putting the list, this figure as 5, so this 5 means more than 75%. More than 75% of the percentage cover of a species. So if the scientist put 5, just only 5, this 5 means that species has a percentage cover between 75 to 95, or 75 to 100%. 4 means, that's between some other figure, like maybe in the 55 and 75, 3 further decrease, and then 1, and then finally 0. 0 definitely means no distributions or no diversity of that a particular species is not evenly present. It's in the same way like you have in Cambridge examination, you have grades. So we say a student have an A plus means it's more than 90%. If it is an A, so it is between 80 and 89. So if a student have a grade A and the other student also have a grade A, we are not aware about their actual marks, which one have the higher marks. Maybe one has 86, one have the 83, but both have the A grade. In the same way, we have some grading system, and that is called abundance scale. Abundance scale. So we have abundance scale 2 and then 1 and then